Ever wish you could like bottle up all the good stuff from a workout, those brain boosting benefits
Yeah.
without actually having to, well, lace up your shoes?
Yeah. Yeah.
Well, you might be closer than you think.
Oh, really?
Today, we're going deep on some really cool research about irisin. It's this molecule your muscles make when you exercise. And get this, it might play a big role in Parkinson's disease.
Wow, that's fascinating.
Yeah.
So we're going to be looking at the study called Irisin, an exercise-inspired molecule for Parkinson's.
Okay.
And our mission today is to pull out the most important pieces of information from this research and figure out what it could mean for how we understand and maybe even how we treat Parkinson's one day.
That's a big one.
Yeah, it is.
Now, when we think about Parkinson's, right, the first things that pop into our heads are those motor symptoms, the tremors, the stiffness.
Right.
But what's becoming clearer and clearer is that there's a lot more going on, kind of behind the scenes.
Under the hood.
Yeah, exactly.
Yeah.
We're talking about neuroinflammation and the loss of those crucial dopamine-producing neurons in the brain.
Right.
And these changes are really important to remember are linked to these abnormal protein clumps called Lewy bodies.
Right.
It's complicated, but neuroinflammation seems to be a really key player.
Definitely.
So the main takeaway here is that tackling this underlying inflammation might be just as important as addressing those motor symptoms.
Absolutely. And it's been really interesting to see more and more evidence showing just how positive exercise can be.
Yeah.
Not just for our muscles and our heart, but for our brains as well.
Right.
Researchers have seen in animal models of Parkinson's that exercise can actually lead to some incredible outcomes, like actually restoring neurogenesis, the creation of new brain cells.
Wow.
And even improving memory and learning.
So it's suggesting there's a much bigger impact than just the physical benefit. Yeah, and this is where things get really interesting. When scientists started digging into the molecular changes that happen during exercise, one molecule kept popping up in higher amounts, irisin.
Huh.
So researchers were faced with this puzzle. Exercise helps the brain, but how?
Right.
And irisin became kind of the prime suspect in this whole biological mystery.
I like the prime suspect.
Yeah. So let's dive a little deeper. What's the connection between this underlying neuroinflammation in Parkinson's and what we know about the disease at its core?
Well, like you said, beyond the motor symptoms, the fundamental problem in Parkinson's is this degeneration and death of dopamine neurons, especially in a part of the brain called the substantia nigra,
Okay.
which is super important for controlling movement.
Right.
We also see these Lewy bodies, which are basically clumps of misfolded proteins, kind of like tangled up balls of yarn that shouldn't be there.
No.
And what's really interesting is that this buildup of a protein called alpha-synuclein, not just in the substantia nigra, but also in other areas like the hippocampus, which is involved in memory, seems to be a major trigger for neuroinflammation.
Okay.
The brain's immune cells, the microglia, they get activated in response to this abnormal protein.
So these immune cells, which are supposed to protect us, can actually become overactive and start causing damage.
Exactly. They release inflammatory substances that can actually further damage those dopamine neurons and contribute to the disease getting worse over time.
Oh, wow.
So if we connect this to the bigger picture, it becomes clear that finding ways to reduce this neuroinflammation could be a really crucial strategy for slowing down or maybe even changing the course of Parkinson's.
That makes sense.
Yeah.
So with all that in mind, what did the researchers find when they looked at exercise in these Parkinson's models?
Right.
What really stood out to you?
Well, the study that involved mice who were exposed to MPTP, which is a neurotoxin that causes Parkinson's symptoms, was super interesting. They had one group of mice just living their normal mouse lives, and then another group who were working out on these tiny treadmills for 10 weeks.
Oh, wow.
And the differences they saw were pretty amazing. The exercising mice showed a clear increase in neurogenesis, which means their brains were making new neurons.
Oh.
And they did way better on memory tests, too, which suggests there's an impact beyond just physical benefits.
So we're talking actual cognitive improvements, not just motor skills.
It's exactly.
It's huge.
And it gets even cooler. They also found lower levels of alpha-synuclein, that problematic protein we talked about in the mice that exercised.
Okay.
And when they looked at cellular markers, basically signs at a cellular level, the markers for inflammation and apoptosis, which is programmed cell death, were moving back towards what you'd see in healthy mice.
Oh.
It really highlighted how powerful the protective effects of exercise can be, hitting multiple aspects of the disease in these models.
So exercise is targeting a bunch of important parts of the disease in these models. And that's where irisin enters the picture, right?
Yes.
What made the researchers zero in on this particular molecule?
Well, out of all the molecular changes that happen during exercise, the fact that irisin consistently increased in such significant amounts really caught their eye.
Okay.
Irisin is this fascinating molecule. It's a peptide hormone mainly released by skeletal muscles during physical activity.
Okay.
It travels through the bloodstream and can affect various tissues and organs, including the brain.
So we know exercise is good and irisin levels go up during exercise.
Right.
But how did they figure out if irisin itself was actually contributing to these brain benefits?
Right.
That's where those more detailed experiments at the cellular level come in, right?
Yes, exactly. They did these in vitro experiments, which basically means experiments done in glass outside of a living organism, like studying cells in a dish.
Okay.
They exposed brain immune cells, specifically microglia, to alpha-synuclein.
Uh-huh.
And as expected, that triggered an inflammatory response. But here's the big finding. When they added irisin to those cells that were reacting to the alpha-synuclein, the inflammation went down significantly.
That's a pretty direct link.
It is.
So irisin was actively calming those brain immune cells in this controlled environment.
Exactly. And they even figured out a specific mechanism. They found that irisin was suppressing NLRP3.
Okay.
Think of NLRP3 like the brain's alarm system for inflammation and a potential troublemaker for the cell's energy supply, which comes from the mitochondria.
Right.
So this was a big step in understanding how irisin might be working its protective magic at a molecular level.
That is super interesting. So they saw this effect in isolated cells.
Yeah.
But how did they confirm that it was really irisin in those exercising animals that was making the difference?
Oh, okay.
That's where the runner serum experiment comes in, right?
Yes, to bridge the gap between those cell culture studies and what happens in a living being.
Okay.
They collected blood serum from rats that had been running on treadmills for 4 weeks. They called it runner serum. When they treated microglia with this serum, they saw the same decrease in inflammation.
Wow.
So this suggested that something in the blood of those exercising animals was having this anti-inflammatory effect on brain immune cells. This is really important because it suggests that the benefits of exercise on the brain aren't just a side effect of being healthy.
Right.
They're actually being actively driven by specific factors in the blood, like irisin.
And then they took it a step further to pinpoint irisin as the key player.
Exactly. They took that runner serum and specifically blocked irisin from doing its thing.
Okay.
And what they found was pretty remarkable. The anti-inflammatory benefits of the serum basically disappeared.
Wow.
This was strong evidence that irisin was a central player in those beneficial effects of exercise on the brain immune cells. It wasn't just some other random thing released during exercise. Irisin seemed to be the key.
That's a crucial piece of the puzzle.
Yeah.
But the million-dollar question is, can irisin on its own without exercise actually provide those benefits in a living animal with Parkinson's-like symptoms?
Right.
What did they find when they gave irisin directly, or in vivo, as scientists say?
This is where the idea of an exercise mimetic really starts to take shape.
Okay.
When they directly gave irisin to mice that had been induced to develop Parkinson's-like symptoms, remember, this is in vivo, meaning inside a living organism.
Right.
They saw all these positive effects that were basically mirroring a lot of what they had seen with actual exercise.
Wow.
They saw less inflammation in the brain, decreased apoptosis, meaning fewer neurons were dying, an increase in neurogenesis, so more new brain cells were being made, and even improvements in cognitive performance on tests for memory and learning.
So it really was mimicking some of those key benefits of physical activity in those animal models.
Yes.
That's amazing. And that brings us to this concept of an exercise mimetic.
Right.
What exactly does that mean, and why is irisin considered such a good candidate for it?
The idea behind an exercise mimetic is to develop a compound, probably a drug or some kind of therapeutic agent that can provide some of the health benefits of physical activity, but without actually having to exercise. This could be really valuable for people who can't exercise because of age disability or conditions like advanced Parkinson's disease.
Right.
And because irisin is a molecule that's released by muscles during exercise and has shown these promising effects on the brain in these preclinical studies, it's become a leading candidate for this kind of mimetic.
Right.
The idea is that by giving irisin or something that boosts its effects, we might be able to tap into some of those brain protective benefits that usually come from physical activity.
That's incredibly exciting, especially for people who have trouble exercising regularly.
It is.
But as the researchers pointed out, we do have to be careful and realistic, right?
Absolutely.
What are some of the limitations and what are the big questions that still need answers?
Well, it's crucial to remember that this research is mainly based on rodent models and cell cultures.
Okay.
While this is a great starting point and it's very encouraging, there are a lot of steps we need to take to translate these findings into safe and effective treatments for humans. We still don't know everything about how irisin works in the human body compared to rodents. There are still questions about the best ways to deliver irisin or increase its production in people, what the right dosage would be, and whether it can provide long-lasting benefits without any unwanted side effects.
And the study also mentioned how exercise has so many different effects on the body beyond just this one molecule.
Exactly.
At-
Exercise is really complex,
Yeah.
and it has all sorts of systemic effects that go way beyond just releasing irisin. It affects cardiovascular, health, metabolism, and the release of tons of other beneficial molecules.
Right.
It's unlikely that any single molecule could perfectly replicate all those benefits.
That makes sense.
So while irisin is promising for its specific effects on neuroinflammation and neuroprotection, it's important to be realistic about its potential as a true exercise in a pill.
But even with those limitations, the potential for helping people who can't exercise easily is still really significant.
Absolutely.
For someone living with Parkinson's or their loved ones, these early findings must offer a glimmer of hope.
They really do. For people with Parkinson's who might struggle with things like mobility balance and just being able to exercise regularly, the possibility of getting even some of those protective effects of exercise through a molecule like irisin is a big deal.
Yeah.
It suggests that we might be able to come up with new treatment strategies that can actually improve their brain health and maybe even slow down this challenging disease, even if they can't physically exercise.
So to sum it all up, irisin, this molecule that our muscles release when we exercise, has shown some really encouraging results in these preclinical studies.
Yes.
It seems like it has the ability to reduce neuroinflammation and slow down that neurological decline in Parkinson's models, basically by mimicking some of the brain-protecting effects of physical activity.
That's a great summary.
Thanks.
It's really important to remember how significant these findings are, but also that this is still early research. The journey from promising results in animal models to proven treatments for people is long and complicated,
Right.
and it needs a lot more research and clinical trials.
So when you think about all the amazing ways our bodies and brains communicate, it makes you wonder what other secrets are hidden within something as simple as moving our bodies.
Yeah.
And what other exercise-inspired molecules might be out there just waiting to be discovered.
That's a great question and something to think about.
Yeah, food for thought.
Definitely.
Well, this has been a fascinating deep dive.
It really has.
Until next time.
See you then.